Welcome. This is Neurobiology Methods, created by Nyantwig Akol. If you've ever wondered, how do we explore neurons? Well, you are in the right place. We all know neurons are information messengers that use electrical impulses and chemical signals to transmit information between different areas of the brain, the body, and the rest of the nervous system. But through evolution and time, how do we continue to explore these neurons? Well, there are 5 main methods or techniques. These include animal models, genetic and molecular techniques, anatomical techniques, recording and manipulating activity, and behavioral analyses. Starting with number 1, animal models. Let's put biology back in neurobiology. The use of animal models has been revolutionary and has allowed us to continue exploring. As we see on the left is the aplysia. Due to the large neurons in the aplysia, scientists are able to investigate and learn more about neurons. And on the left, we have the Drosophila fly. Due to its simplistic neuronal system, scientists were also able to learn more about sensory, motor, and interneurons. Next, moving on to genetic and molecular techniques. Here there are 2 types. We have forward genetics and reverse genetics. Forward genetics allows for the identification and characterization of a gene that is responsible for the mutant phenotype. And we can see that clearly in this diagram with the Drosophila. And on the right-hand side, we have reverse genetics, which, similar to its name, starts in a reverse. We start with a protein or DNA where there is no genetic information, and then working backwards to make a mutant gene and ending up with a mutant phenotype. Next, we have electrophysiological recordings, and there are 3 types: extracellular, intracellular, and whole cell patch recording. In these diagrams, the first one in A, we see the extracellular, and that's because it's on the outside of the cell. In B, we have intracellular, which is on the inside of the cell. Lastly, we have whole cell. This kind of gives us a whole picture, allows for the measurement of the overall electrical properties of a cell membrane, and specifically the total current through all the channels on the membrane and the membrane potential. Next, we have functional calcium imaging, and like the name suggests, this is calcium dependent. This process uses multiple proteins as well as calcium in order to create fluorescent lighting. When calcium is binded to calmodulin and M13, it causes an activation. This activation then causes fluorescent lighting. Optical imaging, which uses light and special properties of photons to obtain detailed images of organs, tissues, and cells, all the way down to our neurons. The cool thing about this is it allows us to see multiple neurons at the same time. Next, my personal favorite, optogenetics, which allows for scientists to control the activity of neurons using light. Light has been proven to be the perfect ligand to open a channel and force the neurons to fire, the channel being channel rhodopsin. This is because neurons don't really care how they reach their action potential. They just want to reach the action potential. It doesn't matter if the channel is a ligand or voltage gated, as long as the medium allows the channel to reach its action potential. And in this case, optogenetics uses light. And as seen in the diagram, shining light at a different frequency does have an impact. Faster flickers equals faster firing. And next we have advances in microscopy. The first one is very well known and often most people have the most interaction with: wide field microscopy. As seen in the diagrams, light shines through everything, and that allows for a nice picture. Next, we have confocal. In this, a laser shines to one plane on one dot and sees a reflection of the light, which cannot be integrated. This is very sharp resolution and a very slow process. Lastly, there is light sheet microscopy, in which light shines through a thin horizontal sheet. In comparison to confocal, this is a very, very fast process, so fast that we can even see developing embryos by using this process. And that's it, guys. Those are 5 techniques used to explore neurons. So next time someone asks you, hey, how do you explore neurons? I hope you can recall this video. All of the citations for the diagrams and the information used can be found in the description. Thanks for watching.